we're really, really fortunate to have Dr. Alicia Martin here to present some of her work today. So Dr. Martin did her undergraduate work at the University of Washington in Seattle, and then went on and did her master's and PhD in the lab of Carlos Bustamante at Stanford University. She moved on to do a postdoc at Mass General, where she remains today after she transitioned to an instructor position and will soon be an assistant professor there at the Mass General. Dr. Martin's work today, she's going to be talking about the ensuring that the translation of these genetic technologies, doing genome-wide association studies and trying to predict complex traits via polygenic risk scores, don't exacerbate health disparities that are induced by the biases in the studies that underlie them, particularly the fact that most of what we know about from genome-wide association studies in humans is largely from white European populations, and that means they have relatively limited utility for looking at other populations. And I want to say, Alicia, I met you for the first time at Biology of Genomes like 5 years ago when you were presenting some of this work, and I thought it was super cool. And so obviously I started following you on Twitter and following a lot of your work, and it's really been a pleasure to see sort of where you've gone, and I love using your material in my classes, as I said. So I'm really happy to have you here. I'm really glad that you said you'd come give a talk. And with that, I'll let you take it from here. I hope everyone will join me in welcoming Dr. Martin.
Awesome. Thank you so much, Noah, for that kind introduction. I'm really excited to have the chance to talk with you all today. It seems like you have such a wonderful department and wonderful set of colleagues. So it seems like such a welcoming community. So thank you very much. Okay, so I'm going to be talking today about sort of a vision for genomics for the world and how we can incorporate a comprehensive framework for genetic studies in diverse populations. So to get started, I'm a geneticist. So as a geneticist, I like to start at the beginning. So for humans, that means we need to start in Africa, where we originated as a species on the order of hundreds of thousands of years ago. And then humans migrated out of Africa and took a subset of genetic variation with them, which is really important to consider when designing genetic studies, because the average out of Africa population has about 1 million fewer genetic variants than the average African population. So this human history over the course of the past hundreds of thousands of years informs the landscape of genetic variation across the world and is really important to consider when we're starting to think about the genetic basis of evolutionarily important traits and different biomedical diseases. So I think it's probably the most exciting possible time to be working in human genetics because of how rapidly and explosively our studies have increased in size. So looking over the course of the past decade and a half or so, our studies grew from in 2006 on the order of hundreds or maybe thousands of individuals to now routinely having millions of individuals in these very large scale genome-wide association studies. So with this really vast explosion in these studies, we've learned a ton about human history, but also about the biomedical basis of disease from this exponential growth. So I think this really serves the mission of genetics very well, which I see as providing biological hypotheses from unbiased associations, which then fuels the basis for downstream functional analysis and then eventually, hopefully, therapeutic insights. But looking at this chart again, we can really, I think, easily recognize that genetics has a huge diversity problem. So looking at this chart now plotted by population, you can see that the vast majority of participants in these big genome-wide association studies, or GWAS, are European ancestry populations, which is far out of step with the global population. So right now, about 80% of participants in these studies are of European descent, whereas about 16% of the global population is European or European descent. Perhaps even more problematically, though, if you look at the bottom part of this graph, you can see that the non-European fraction of participants in these studies peaked around 2014 or so, and since then has pretty much stalled or slid in the wrong direction for diversifying progress. So this is really happening because our European ancestry studies keep getting larger and larger and larger, whereas our non-European ancestry populations, which constitute the majority of the globe, are staying pretty much the same and haven't really grown at the same scale. So this is a really huge issue. It's starting to change, but we've been saying it's starting to change for the past decade or so. So this, I think, really needs to have rapid transformative progress if we want to understand the genetic basis of disease, because to fundamentally understand the genetic basis of disease, we need genetic variation. So if we have blinders on and are looking at less genetic variation due to this more narrower focus, then we're bound to find fewer genetic associations. This is not a new problem. Obviously, this has been a big issue for a long time, not just in genetics, but in science more broadly and in society more broadly. So we've been working to correct some of these issues in genetics, but progress has really been quite glacial compared to how rapidly some of our studies have grown. So I think it's important to reflect, particularly in this crazy year of 2020, how pivotal this time is, and recognize how institutionalized and ingrained these issues are and why they've come to be. So clearly, some of the leading journals in science today have really recognized that science has a racism problem and that we need to really fight against these issues to transform leadership and to transform the diversity of our studies. So as I mentioned, 2020 has been a year of reckoning. It's forced us all to evaluate what's important and what matters most to us, both in our personal lives and in our work lives. So I've been starting a lab this year, perhaps in some ways luckily, in some ways less luckily, but one way that I feel like I've been really lucky is that I've been starting a group while reflecting and prioritizing which areas have the biggest needs and can provide the biggest opportunities. So the goal of my work is to facilitate diverse population genetic studies to ensure health equity, or to ensure that the translation of these genetic technologies performs equitably across different populations. So we have some really big gaps in the field of genetics that can't be solved by one lab or one group, but that needs to be solved at the field-wide level. And with these needs met, there's some massive scientific and ethical opportunities downstream that will follow from these. So on the needs side, we need better statistical methods. Most of our methods that are being used assume some degree of homogeneity in our genetic studies. So we need to start developing methods that can take into account more heterogeneous, higher levels of genetic diversity to conduct large-scale studies. We also need better supporting data and community resources. So some of our biggest resources already have large-scale, unanalyzed data sets of primarily non-European ancestry populations, but that aren't really being analyzed. Additionally, we've had a really big head start in a lot of areas that have really focused on these vast Eurocentric cohorts that have really built up these big collaboration networks. But we need to start building far beyond those collaboration networks and starting to collate data beyond these boundaries. And then, of course, the existing data alone is not going to solve issues of Eurocentric study biases. So we need to grow and develop new data to address these gaps. And then we also need to develop supporting resources like imputation panels, cell lines, oops, sorry for the typo there, and other factors. And then we're never going to be able to make sufficient progress without ensuring that our research capacity is building to train the next generation of researchers in diverse communities and diverse countries to start to lead these efforts on the ground. So with all of these kind of needs being met, then there are huge scientific opportunities. So one is we can identify new genetic variation. There's a lot of low-hanging fruit that we've begun to learn is out there and that we can identify by diversifying our studies. Additionally, fine mapping presents a unique opportunity in increasingly diverse genetic studies because we can leverage this very diverse population history to more accurately pinpoint and increase our resolution to find causal genetic variants that'll then accelerate our functional follow-up studies by making them more efficient. Genetic risk prediction is an area that I'll spend a fair bit of time talking about, so I won't spend too much time mentioning that here. And then lastly, genetic and environmental determinants of health are not necessarily shared across different populations. So we need to diversify our studies to understand which factors are shared and which population risk factors are unique. So I'm breaking this talk up into 3 sections. The first section is going to talk about diversity being critical for equitable translation of genetic studies. The second section will talk about multi-ethnic studies and how uniquely informative they are for genetic and environmental determinants of health. And then the last section, I'll spend a bit longer on talking about where do we go from here? How do we facilitate diverse population genetic studies? This is really where that comprehensive framework idea comes in, which is kind of going to serve a little bit as a call to action for everybody to kind of work together to increase diversity in our field. Okay, so diving into the first section, one of the core and central questions of my work is how do ancestry study biases in genetics impact the generalizability of our knowledge? So there's a couple of important considerations to keep in mind when trying to answer this very broad and very big picture question. From a genetics perspective, we really fundamentally expect the biology to be shared across populations. Fundamentally, the molecular pathways, the cells, the processes that are involved in various diseases and traits are expected to be mostly shared across populations. This shouldn't come as a surprise. The reason someone will get a heart attack in one population compared to another is likely due to the same molecular pathways. But we expect that different genetic variants are going to be shaped by our evolutionary history. So allele frequencies, linkage disequilibrium, our environment, and other complex factors are likely to shape what we're learning from these large genetic studies, even if the causal underlying biology is shared, because these forces impact what we can estimate and what we can learn from these studies. We're not getting causal effects, we're estimating these effects. Correlation is not causation is the, I guess, common mantra here. So as a tool to try to understand this generalizability, I've been using a polygenic risk score, which I'm guessing many of you have likely heard of in the past because it's been sort of bandied about quite a bit in the popular media. So a polygenic risk score has a pretty simple underlying concept. This is fundamentally just the genetic prediction of an individual's phenotype. So to construct a polygenic score, we assume that a genetic study has been conducted before. They've estimated effect sizes for millions of SNPs across the genome. And then we have an independent target data set. So we have this, these effect size estimates from an existing study. We have this independent target set where we have some genetic variants, and we're trying to predict in this independent target set. So to do that, we basically take the effect sizes estimated in this previous study, we multiply those by the genotypes in our target cohort, and then we sum those up across the genome. And basically that gives us our genetic prediction score. This is a pretty simple concept. There's a lot of kind of nuances and different methods that you can use to construct these. But the fundamental idea is that we have a polygenic backbone. Most of the time when we're looking at complex diseases and traits that we're interested in, like say heart attack risk or type 2 diabetes risk, or even things like autoimmune disease risk, we're looking at diseases that have risk predicted by more than just one genetic variant in the genome. In fact, more of the genome than not seems to be contributing. So polygenic scores have been on people's radar, I think, especially lately. And so there's been a lot of hype about them and then some sort of tempering of that hype. And I just want to walk through a few scenarios that people have kind of been playing out as their potential implementation and use cases. So the first area that people have been talking about polygenic scores is in research. So we're already actively using polygenic risk scores in research. But there's a lot of places where this might be more useful than others. So for example, I work especially on psychiatric disorders, as one of my affiliations is at the Stanley Center for Psychiatric Research. And so here in this area, you might think of something like schizophrenia as a disease area of interest. But it's not necessarily useful for me to predict whether or not someone has schizophrenia because they're standing right in front of me, and often it's relatively straightforward to, or well, less so in psychiatry, but in other disease areas, there's often diagnostic tools that can tell you whether or not someone has a disease. But there's a lot of downstream consequences of diseases and disorders that are very helpful to understand. So with these polygenic scores, perhaps you can use these to try to understand whether someone's going to respond to one drug or another better or not, and how likely are they to be readmitted to a hospital? What other sorts of downstream consequences might someone be at risk for if they have a particular disease? This has also been talked about in the context of clinical trials and has started to be taken up in this context. So the idea here is that genetics and environment both play a role in whether someone will have a disease. Those with a high genetic predisposition may have unique biological pathways that are at play and that are potentially at risk in high risk individuals. And so, if we can identify those individuals at high genetic risk, perhaps that can accelerate the efficiency of clinical trials. And these are incredibly expensive. So, any degree of improvement is likely to translate into a large cost benefit and cost savings. And then the last area that people are talking about, I think, with the most enthusiasm is in routine clinical use. So differential diagnosis, I think, is not an area where this will have a big impact because oftentimes the disease areas where you might be interested in differential diagnosis, say something like schizophrenia versus bipolar disorder, these are actually really genetically correlated. And because of that, there's not going to be much utility in using genetics to try to discriminate two diagnoses. However, people are talking about this a lot in the context of preventative medicine. So you could potentially use a polygenic risk score to predict whether someone is going to go on and have a disease in conjunction with other clinical risk factors and environmental risk factors. Right now, our studies tend to be somewhat simplistic and just look at polygenic scores alone. But you could imagine a world in which we're incorporating these other clinical risk factors, like is used in heart disease all the time, looking at things like BMI, smoking, hypertension, cholesterol, in addition to a polygenic score, as well as maybe what kinds of food security does a person have and what kinds of access to other socioeconomic factors are they disposed to. So with these in mind, we were really interested in this idea of the generalizability question. And so coupling population genetics with statistical and complex trait genetics, we performed a few years ago now a simulation-based and somewhat empirical-based study to try to understand this generalizability question. So we set up simulations using a demographic model of population history that was purely neutral, so no selection was applied here. And what we found from this study, I'll go through a few of these points, was that the first one was that genetic prediction accuracy decays with increasing genetic divergence between the discovery cohort and the target cohort. So if I have a large cohort of European ancestry individuals, the more genetically distant someone's ancestors are from those European cohorts, the less accurately I'm going to predict their genetic risk. That's maybe somewhat intuitive. The second thing that we learned is that by applying these polygenic scores in data sets that are just publicly available, like the 1,000 genomes cohort, for example, using GWAS that have been conducted before, we learned that polygenic scores differ across populations arbitrarily often, and these biases are not necessarily meaningful. So to give an example, I'm showing on the side of this slide a polygenic score for height in different populations from the 1,000 Genomes project. And so here you can see that European populations are predicted to be taller than South Asian and admixed American populations, and that East Asian populations and African populations are predicted to be the shortest. And these differences in these distributions are extremely non-trivial. So there's like 5 standard deviations worth of difference between these European and African populations, for example. And so if the average European ancestry man is, say, 5 feet 9 inches tall, and the standard deviation for their height tends to be about 2 or 3 inches, then this 5 standard deviation difference would peg the average, say, African man to be shorter than me. And we're not in the same room together, but I'm about 5 feet tall. So that's clearly out of step with observation and reality. So we learned from this that some neutral human evolution is sufficient to explain these differences. That doesn't rule out the fact that selection can be at play, but these biases are not necessarily driven by selection. So following up on this work, some researchers in my environment and I collaborated and worked on this study to identify polygenic scores across a more globally set of populations using GWAS data from an aptly named GIANT consortium. So this looked at, this was a GWAS study of height conducted in about 250,000 people, primarily using European ancestry populations, but spread across that breadth. And so using these polygenic scores for height, using the same data set and then applying them in a broader set of populations, they recapitulated many of our same findings, which is that you expect from these data that European ancestry populations are taller than, say, African and East Asian populations. So that's what they found using the giant data. But moving forward and using the larger data set that was available at the time, the UK Biobank, they saw that a lot of these differences disappeared. So these studies were slightly different in size. Giant had about 250,000 people. The UK Biobank data that's being analyzed here included about 350,000 people. But really these differences are enormous between these two different analyses. So it's important to recognize how these data sets were constructed. So Giant was constructed by meta-analysis. So taking many small cohorts and putting them all together through this process of meta-analysis. So they were collected in different times in different places, and then the statistics were just aggregated. Whereas the UK Biobank was conducted in a very harmonious way. Everything was sort of put together all in the same nice place. So it was very easy to control for population structure. So one hypothesis was that maybe this harmonious structure of these data sets was actually a good way of getting around this residual population stratification, which we thought was inflating polygenic score differences across different populations. That would be really nice because it would give us a potential solution of just making sure that our studies are more harmonious and are sharing individual level genetic and phenotypic data. Unfortunately, we later learned that that's not sufficient to control for these differences, and that residual population structure continues to be an issue when applying polygenic scores. So I teamed up with a talented grad student, Siini Kermanen, in the Finnish Institute for Molecular Medicine. We looked at polygenic scores in Finland, partially because Finland is an ideal test case. So they have an excellent integrated healthcare system. About 10% of the country is on their way to being genotyped with integrated health records associated with that genotype data. And there's a lot of social trust in their healthcare system. And there's the potential that polygenic scores could be implemented in practice in the relatively near future. So when we looked at height, one of the other motivations for looking at this in Finland was also that there's clear empirical evidence of height differences across the country. So we see from the southwest to northeast parts of the country that there's an observed 1.6 centimeter difference on average. So we already knew that there was an empirical difference in height. So we used multiple different GWAS data sets to then try to look at what polygenic scores for height looked like across the country using the giant data, the UK Biobank data again, and then this time also adding in a data from Finland. It's smaller than these other two data sets, but was collected in a similar way to the UK Biobank. So it's all harmonized and nicely processed together. It includes about 25,000 people. So we recapitulate what was seen before. With the giant data, we see that this is explaining about 14% of the phenotypic variation, but is already predicting a difference of 3.5 centimeters across the country. So over predicting the differences substantially, even though it's explaining only a fraction of this variation. The UK Biobank, it's a larger study, so it's explaining more of the phenotypic variation, about 22%, and it's predicting a difference from the eastern and western parts of the country of about 0.6 centimeters. So this is about on track with what we would actually expect. But the Finnish data set, which is much smaller, about 25,000 individuals, is explaining about the same amount of the phenotypic variation as the giant study, which is maybe a reflection of the fact that a study that's done in a more ancestrally similar population can explain more of the phenotypic variation. But here we're only explaining 15% of the variation and yet predicting basically the full east-west observed difference. So again, we're observing this over-predicted height difference in Finland. So these are kind of silly examples of height. We can just kind of look at a person and know whether we're over predicting a difference or not. But if we're thinking about this in the context of healthcare, of course, this can be a big issue because if we're predicting something like heart attack risk, and you just inherently assume that one part of the country is going to have a lower risk of heart attack, then you might do less screening, which is clearly going to be a big issue and lead to negative health outcomes. So I do think polygenic scores are exciting as a potential biomarker. I can't think of a single lab test that can be so predictive of such a broad spectrum of disease. However, one of the biggest challenges, I think, to implementation of genetics and precision medicine is the fact that our genetic studies are so Eurocentric. So we wanted to understand how polygenic scores are performing in ancestrally diverse populations. So here we turn to the UK Biobank, which I've mentioned now a few times. This is a collection of about 500,000 Brits living in the UK and who volunteered and signed up for this study that really systematically surveyed their health in a really unique way, and then provide a pretty open resource to researchers to dive in and try to understand the epidemiology of many different health traits and phenotypes. So when we looked at polygenic scores for 17 different quantitative traits, things like height, BMI, and other blood panel traits, all continuous things, we were able to predict traits in European ancestry populations far more accurately than elsewhere. So for example, we're doing about twice as well at predicting traits in European ancestry populations compared to East Asian populations, ancestry populations living in the UK, and about 4 to 5-fold as accurately as in African ancestry populations. So we expect fundamentally that the causal variance and the causal pathways are shared, but we understand that the genetic basis of these differences is due to differences in correlations of these variants, differences in allele frequency, differences in LD. So getting down to some of these problems, we understand that prediction accuracy decays with genetic distance, but why? What's really driving this? So one of the first issues is that these large genetic studies are best powered to discover variants that are most common in the populations that we're looking at. So if we keep studying European ancestry populations over and over again, we're bound to find genetic variants that are most common in those European ancestry populations than elsewhere. That means that there's a lot of low-hanging fruit that's polymorphic outside Europeans, but remains fixed in Europeans to be discovered. So as an example here on the right, I'm showing you this very canonical and clear, biological phenomenon, where if we just look at a genetic association study using white British samples from the UK Biobank, and then compare that to what we find when we run a GWAS using the multi-ethnic and much more ancestrally diverse set of participants from the UK Biobank. When we're looking at this trait, mean corpuscular hemoglobin concentration, or hemoglobin, we're identifying in the pan-ancestry analysis unique variants like sickle cell alleles and beta thalassemia variants, these very canonical things that are really important for predicting this trait in these underrepresented populations, but that we wouldn't find if we just studied the European ancestry subset. So the next component that makes this difficult is that there are LD differences across populations. So I'm showing on the bottom right part this LD structure between pairs of SNPs. So the closer you get to the diagonal, the closer you're looking in the genome in proximity. And the further you get from that diagonal, the more distant SNPs are from each other, the pairs of SNPs are from each other. On the bottom left part, you're seeing LD in the European ancestry population, and in the top right part, you're seeing LD in an East Asian ancestry population. And so by and large, you see symmetry across this diagonal, indicating that LD mostly has similar structure across these populations, but I'm highlighting a region where you can see extended LD in the East Asian ancestry populations that's not present in European ancestry populations. So when we have regions where we have this extended LD or differences in LD across populations, our effect size estimates from GWAS turn out to be different, because even if our causal variants are the same, what we're doing our association test on is not just the causal variants, but also the correlation or proximity to those causal effects. And then lastly, there's all sorts of complicated environmental, selection, and other complicated differences that I don't have time to get into today, but there's a whole rich literature that's just sort of outpouring right now on the effects of all of these complex differences. So the next part of this is going to be a quick section on how multi-ethnic studies are uniquely informative of genetic and environmental determinants of health. So I've talked a little bit about the UK Biobank already. This is a cohort that's a general population cohort. People volunteered for this study. This volunteer effect means that people are healthier, wealthier, and higher educated on average than the general UK population. We wanted to compare this to the Biobank Japan to try to understand how similar or different across different cohorts our polygenic scores is our generalizability across these groups. So the Biobank Japan is different in that it's a hospital-based study, and that it recruited across 47 different disease areas. So this ascertainment on these 47 different disease areas means that participants in the Biobank Japan tend to be less healthy on average than the entire country's population. So we wanted to evaluate the promise in these diversifying efforts. So if we have multiple different ancestrally diverse sets of cohorts, how are we doing in our discovery and in our polygenic prediction? So I teamed up with this incredibly talented student, Masa Kanai, who's worked with the Biobank Japan data for a long time. And so we wanted to see if there were symmetric and comparable polygenic score accuracy differences if we set up our studies in the most symmetric way we could think of possible. So to do this, we ran equal sized GWAS for the 17 different traits that I had talked about before, as well as 5 different diseases in UK Biobank and in Biobank Japan. These GWAS were on the order of 80,000 to 150,000 individuals. And then we computed both within and across population polygenic scores. So I don't have time to go through all of the results, but I think you have access to this paper and it may have been reading from before. But some of the key takeaways here are that our ancestry matched results are always performing best when we're doing our polygenic prediction, but our most accurate results are coming from the largest meta-analysis of the most diverse set of populations that we have access to. Another finding was that heritability differs by cohort. This is not like a fixed entity. If you ask me what the heritability for height is, the question is going to be in who, at what time, in which cohort. It's all fairly specific. The heritability of height in infants, for example, is going to be different from the heritability of height in adults. And part of this comes also down to phenotype precision. So I mentioned before that Biobank Japan was particularly focused on disease areas. So their phenotype definitions for diseases were more accurate than in UK Biobank. And conversely, UK Biobank had higher heritability estimates for all of the quantitative traits that we were looking at that are shown sort of along the slide. And so those are really big, important factors to consider. Okay, so now where do we go from here? Hopefully I've convinced you that it's really critical that we start to deeply invest in diverse population genetic studies for all of our broad efforts and goals in the community. So there are 4 different areas of scientific objectives that I want to go through that we're starting to tackle, but others are very keenly involved in making major contributions to these. So the first is in genetic discovery and analysis efforts, especially in polygenic risk scores, using large-scale, diverse population studies, especially in biobanks. The next area is in building large-scale community resources, also in developing statistical methods and in advancing research capacity. So there are 4 big projects that I'll just outline and go through briefly that I'm super excited about and motivated by. This is a big picture overview of all of these, and I'll dive into them in a little bit more detail. So NeuroGap, or the Neuropsychiatric Genetics an African population study, is one of the most rewarding projects that I've ever been involved in. This is a major effort that's enrolling 35,000 participants, about half are cases, about half controls. That's already over halfway through enrollment and recruitment into the study. That's looking at the genetic basis of psychosis in Ethiopia, Kenya, Uganda, and South Africa. So a study this large happening across this many different countries hasn't really been conducted before at this massive level, which has brought really amazing, unique opportunities, I think, for collaboration across these institutions, and has also meant that we've had to go discuss, for example, with the ministers of health and make sure that they're all on board with conducting a study of this size. And I should mention that a lot of this effort needs to take place in these countries and is being led by local investigators. The second project is the Populations Underrepresented in Mental Illness Association Studies, or PUMAs, in which we're sequencing to low coverage about 120,000 African and Latin American populations. About 40,000 will be schizophrenia cases, about 40,000 will be bipolar cases, and about 40,000 will be controls. So this is going to be an enormous study that'll take place over the course of the next 5 years. It was funded earlier this year, and we're so incredibly excited about, and will be sort of unprecedented in scale across these different countries. The next area to mention is the Pan-UK Biobank project. As I mentioned earlier, most of the time when people are analyzing data from the UK Biobank, they're focusing primarily on the white British samples or the European ancestry samples. But there are participants in here from all over the world with ancestry coming from all over the place. So we've expanded the analysis of this data set and released this out to the community to ensure that people are making better and more diverse use of this massive resource. So we've run over 16,000 GWAS across 7,000 different phenotypes in 6 different continental ancestry populations. And then lastly, I'll probably skip over for time's sake, but just mention the Global Biobank Meta-Analysis Initiative, which is bringing together biobanks across the world to increase discovery and prediction, particularly for understudied populations where there are known substantial disparities across populations. So I mentioned the PUMAS project. This is a huge project that includes multiple different participating sites. So this is an umbrella project that encapsulates NeuroGap, which I mentioned a little bit before, as well as several different other projects that include colleagues at the Harvard School of Public Health, SUNY Downstate in New York, and UCLA, as well as the Universidad de Antioquia in Medellin, Colombia. So this is taking place across several different countries in Africa and Latin America, and we'll be building up this sample bank of 180,000 participants and then doing sequencing on a set of 120,000 of these. And so the major goals and questions that we're trying to answer in this project are, what are the genetic variants contributing to mental illness, particularly for schizophrenia and bipolar disorder? And when we can find map these causal variants, are they shared or are some of them population specific? Are we seeing an allelic series for these variants? And how much heterogeneity is there actually across these different populations when looking at the genetic basis of these traits? Also, it's going to be a huge harmonization effort, bringing all these different phenotypes together. So also, how consistent is our different phenotyping strategy in these different countries? So the Pan-UK Biobank project that I mentioned, this is a huge effort that started over a year ago when another instructor and I got together, Elizabeth Atkinson and I were working, and we realized that several of the ancestry groups that are included in the UK Biobank weren't being analyzed, but were some of the largest that were available for a given phenotype, even if they're a tiny fraction of the UK Biobank cohort overall. So for example, this whole cohort is about 500,000 people, but there are still over 20,000 individuals coming from these primarily non-European ancestry groups. So we pulled together a pretty big effort and did a reanalysis of the UK Biobank spanning all of these different phenotype areas. So we looked at continuous traits, biomarkers, categorical traits, ICD codes, so things like electronic health record data, aggregations of these ICD codes, like heart attack risk, for example, and Phe codes, and then prescription data, which was new to this. So we released about 12 terabytes worth of summary statistics earlier this summer. This is a massive resource. I wouldn't recommend downloading all of it, but we've also made available each of these sets of summary statistics earlier this summer that you can download if you're particularly interested in a given trait. I've listed the website on this slide in case you'd like to go check it out. We've also released a set of frequently asked questions, or actually anticipated questions that we would get. So we put together about 17 pages of content that were targeted at lay and scientifically inclined audiences, describing the risks, benefits, and limitations of this resource, anticipating that when providing genetic studies from multiple different ancestry groups, that this was sort of a potential issue and that people may misinterpret some of the findings when comparing across groups. We also updated the statistical methods that we're using to include generalized linear mixed models implemented in SAGE. So this is sort of more robust methodology for multi-ethnic and multi-ancestry groups compared to what was previously used, which was just straight linear regression with including some principal components. I mentioned earlier that many phenotypes are new here, including Phe codes, prescription data. We've also been continually analyzing and updating COVID-19 diagnoses and death rates, and releasing this as and contributing this to the COVID-19 Host Genetics Initiative, which is aggregating across many different genetic studies. Clearly, your likelihood of getting COVID is environment-based and just exposure-based. But if there are particular genetic variants that contribute to how you respond to this virus, that's of particular interest. So we have many different analyses underway, looking at new biology effective gains in power from this increased diversity. So as I throw 10,000 new individuals into the analysis from a diverse ancestry group that's primarily from a non-European ancestry background, what's the power gain that I have compared to if I just added 10,000 more Europeans to the group? We're also looking at prediction power increases. So in polygenic scores, for example, we're asking how much of the gap are we closing by increasing diversity in our GWAS data sets. And then we're also comparing within and across different populations, looking at disease risk prediction as a function of both polygenic scores and environmental variables. So not doing the simplistic thing of just how well am I able to predict a disease from a genetic risk component, but how well am I able to predict disease as a function of genetics and environmental factors from this deep epidemiological cohort. I'll skip over this for now, just for the sake of time, so that we have a chance to get to other discussion points. But just wanted to mention that GBMI is bringing together biobanks from all across the world at the scale of millions of individuals. And there's really unique opportunities to dive in here, particularly in understudied areas. So with all of these amazing analysis opportunities and these new genetic discovery cohorts, we need commensurate community resources. So we kind of need to bring our public data sets up to 2020 speed, or maybe hopefully 2021 speed. So most people are still using the 2015 release of sort of medium coverage 1,000 genomes data to use as a population reference for example, imputation in GWAS or in comparative population genetic studies. But we now have deep 30x whole genome sequencing that's on the latest human genome build of GRCH38 for both the 1,000 Genomes project as well as from the Human Genome Diversity Project. So we've brought in these data sets that have been sequenced by external additional wonderful colleagues in the field, for example, at the New York Genome Center and at the Sanger Institute. And we've partnered with the Nomad team, the genome aggregation database team, to integrate these into this wonderful resource to ensure that we can start to build this out and that people can access high quality individual level genetic data for whatever projects they're interested in using this for. So this builds upon the previous 1,000 Genomes release, for example, by including populations that were previously unrepresented in public data sets, including public sequence data sets, including the Middle East, as well as vast swaths of Africa. This resource includes about 220 million genetic variants. Compare that to the previous releases of 1,000 Genomes that included something on the order of 90 million genetic variants, highlights really the vast diversity present in these data sets. So we're planning on integrating this into future public data sets used in tutorials and reference panels, things that can be used for imputation or just as population genetic databases. If you want to download and start playing around with this data set, we have some work underway on this, but you're totally free and welcome to access this and start playing around with it. There's a link on the bottom slide. We're also developing new statistical methods for diverse population analysis. We're particularly focused on multi-ethnic meta-analysis, as well as multi-ancestry polygenic risk scores. We've got some ideas about how to bring together also multi-trait and multi-ancestry methods. So you might anticipate that you'll have genetic study results available for something like schizophrenia, as well as bipolar disorder in European ancestry populations, as well as in, say, East Asian ancestry populations. So doing these really large scale analyses requires supporting methods that are aligned with our goals. So when we're doing genetic risk prediction right now, it's always a question when we're trying to implement these methods, what do we do in diverse, admixed, and heterogeneous populations? We're hoping to build out these statistical methods so that this isn't a question, there's just a clear method that you can use. And it's really important that these start to get implemented super rapidly because polygenic scores are already going into clinical trials. So we need robust approaches and methods like yesterday. So one method that we're working on in our group is the multi-ancestry meta-analysis method, which we call MAMA. This approach assumes that you have GWAS data from 2 or more different populations and that you're interested in meta-analyzing across these groups. Current inverse variance weighted meta-analysis usually has biased effect size estimates. So here we're adjusting across populations using LD information, both within as well as across populations to recalibrate effect size estimates in an unbiased way. I won't go into this too deeply. So we also really need to build up our research capacity. Over the past several years, we've been doing this through a program called the Global Initiative for Neuropsychiatric Genetics Education and Research, which we just call GINGER for short for obvious reasons. And this has been a really wonderful program that's been deeply and integrated into the NeuroGap program. So there are 17 research fellows that are sort of rising stars in the statistical genetics and population genetics space that have been involved in workshops, that have been going through online virtual weekly classrooms, learning about statistics and genetics. And then we've also tailored on-site training in which they've served as TAs to build up a community around them. So they're not, for example, the only ones doing GWAS that know how to do this sort of thing. So the first iteration of this program was 3 years. I was really lucky to work with 3 of the Ginger Fellows really closely. And we have a project that's wrapping up now that they've been just, they've learned so much, and it's been wonderful to watch their progression. So the first iteration was 17 research fellows for 3 years. We've fortunately been funded for another 5 years to have a second iteration of this program that will involve 7 research fellows that'll have a more immersive and longer term growth progress. I'm going to skip over my own lab stuff and just focus on what the Ginger Fellows have accomplished. So I've worked particularly closely with Lorato, Nastasia, and Alan, who have driven a project looking at the transferability of polygenic risk scores across diverse African populations. So Africa is a hugely diverse continent, different populations across different regions of Africa are as differentiated or as diverse as populations across different continents out of Africa. So we've looked at polygenic score accuracy across these different groups and have seen that within Africa, as expected, polygenic score accuracy across some of these groups is as different as across some out of Africa continents. I'm just going to skip one of these findings, but stay tuned. There's going to be a preprint coming on some of the really wonderful work they've done probably in the next couple of weeks. Okay, so for next steps, I see polygenic scores as potentially providing an objective biomarker and improving clinical models, but right now, I think they're likely to increase health disparities due to these vast Eurocentric genetic study biases. And stepping back and thinking about the field more broadly, we really need to ensure that this doesn't happen by diversifying our GWAS, developing new methods, building new resources, and increasing our research capacity to address these major issues. And as we do so, I think it's really important to anticipate the implications of our research. This requires a level of nuance that is not necessarily always the case in science. So I think it's really critical that we communicate these culturally sensitive topics responsibly and widely. So with that, I'll say thanks. I'm happy to take any questions.
Sometimes it feels like you're talking into a void, but trust me, you're not. That was really, really, really wonderful. Thank you so much. And it's a great way to wrap up the semester. I'm going to open up for questions here. If anyone wants to type in their questions or raise their hands, ask something out loud. I have we've got a few questions, and I can always ask you questions afterwards. So, Aaron first asked the question, basically thinking about like, how can you apply this to other traits? I don't know if you can see his 2 questions there, and the other trait that he's talking about is suicide risk or suicide itself, suicide-related behavior. Have there been GWAS and polygenic risk scores done for those things?
I think there have. It's not as much my area, but one area that I have looked a little bit at is in PTSD. I have some close collaborators that work there, and they certainly have looked at polygenic scores there and have seen that, many traits that we look at tend to be somewhat predictive, for various reasons. So we can look at, cardiovascular disease risk and see that it's heritable. We can look at PTSD and see that it's heritable. We can look at suicide risk, and I would guess we would also likely see that it's heritable. There are various reasons for this, and sometimes when looking at things like, this starts to get a little bit touchier and an issue of what makes suicide risk heritable. Perhaps this might be related to things like depression, but this really starts to touch a little bit on social genomics to some extent, which is always an issue that I want to couch in a way that is like aware of the fact that genetics is not sort of a be all end all, and that there's a lot more often that's not genetic that contributes to these traits than is genetic.
Yeah, that's a great answer. And you sort of touched on that in your talk when you said the genetic and environmental determinants of health, which is really important to think about. I want to just jump off that a little bit more and say, okay, so you were talking about another phenotype. What if some of these phenotypes aren't measured or haven't been measured or can't really be measured, or are harder to define in some other populations? I can't think of an example off the top of my head, or maybe due to stigma or something like that, they're not diagnosing some of these. Is there still some utility of polygenic risk scores? Like, would you develop it in like a as closely related of a population, and then would that have some utility in predicting? I mean, you couldn't predict like whether or not they have something because it can't be diagnosed, but you could put them on some sort of risk spectrum and then target interventions or something. I hope that was clear.
No, that is clear. And it's also, I think we actually have like a really clear example of that. So in the Stanley Center, we're particularly interested in psychiatric disorders and mental health and wellness. That mental health in general comes with a lot of stigma, particularly when there are psychiatric diagnoses in East Asia, for example. We have a ton of colleagues who are trying their best to get diagnoses at large scale that are willing to be shared in different parts, in different countries, in different regions, for example, of China or different regions of Japan. And it's actually really hard to get these large scale biobanks, these large scale data sets available. So I mentioned Biobank Japan, and so there are 47 different disease areas that they're ascertaining on. Mental health is not one of them, and there's a reason for that. It's stigmatized to the extent that it's just not easy to share that kind of information. So I think that this could be very useful in that context, as well as like providing for stigmatized traits some information where it's not available. Although, again, I would like to couch that in genetics is not everything, and for stigmatized traits in particular, having some genetic predisposition for risk is potentially culturally challenging. There was a huge example of this, I think, last year when there was a genetic study of non-heterosexual behavior. And then there were downstream consequences to this type of work where, for example, this is punishable in very, very severe ways in some parts of the world. And there was a genetics app predicting how likely you were to be gay, for example, in Uganda, which is not something you want available in Uganda or out there. So I think we really need to be careful about what we put out there, but there is also some value when you are starting from a ground zero point for some of these traits. So it's just tricky.
Yeah, that's a really tough needle to thread, and like using the Ugandan example is a really good one. I mean, thinking about how well you can predict one individual with a polygenic risk score, and it's usually not that well, and right overall, I would say. And so,
yeah, absolutely.
Can you expand on that maybe a little bit more? Because that's something I think that exactly like maybe the Ugandan government is like, yes, now we can genotype people and tell whether or not they're homosexual, and we should punish them by death. Like, why is that? Like, that's just unacceptable and gross. But like, why is that like theoretically and scientifically not sound either?
Yeah. In addition to all of the social and very, very, very big problems that come along with it, the science is also not even feasible for like an individual level polygenic score for these complex traits. We're doing genetic studies where we're finding thousands or sometimes tens or thousands or hundreds of thousands of genetic variants that are contributing to a trait, and these are not definitive markers. These polygenic scores are also useful at the population level, not at the individual level. So if we can predict risk at a population level and decide to put more people into screening programs for particular disease areas that are actionable, say like breast cancer or cardiovascular disease or those kinds of disease areas, that's going to be useful and meaningful at the population level, but often not at the individual level, because it's not that we're taking an action that's meaningful to that person individually usually, but more so at the broad screening level.
I'm going to try and read through some of these questions here because I haven't done it too. So Lucas said he started reading the paper about how genetics research is geared towards white European populations, immediately jumped to sickle cell anemia, which is far more frequent in Black newborns than white newborns. And this is more hypothetical, but if the roles were reversed where white people were more likely to be affected by some of these diseases, what do you think would, where do you suppose we would be in regards to this? Like, do you think we would have developed more tools for, I don't know, lots of things related to health and genetics?
Yeah, it's a good question. I would say that we've known about sickle cell for a long time. I guess another disease area that's kind of analogous a little bit is it's a Mendelian disease. So things like cystic fibrosis have also been known about for a long time. And we're only just now getting to the point where genetics is actually coming to fruition for drugs development. So I do think we would probably be in a different place, but the technologies have been developing over the course of the past decade or so, and the genetic technologies are now being translated, hopefully, I think for a broad spectrum of diseases. So it's hard to say. I don't have like an alternate universe cap that I can easily put on, but I'm happy that some of the genetic technologies are helping cure diseases at all. And I hope sickle cell is like up there next for, just rapid, curing basically.
I imagine in one of the other multiverses, you are giving a talk that is just about that right now.
Yeah.
So Randy Nethie asked the question in a 2018 article by Curtis in Psychiatric Genetics, found that the polygenic scores for schizophrenia varied 10 times more between groups of different ancestry than they did between schizophrenics and controls. What do you think this means for schizophrenia genetics and the possibility of finding allele specific to schizophrenia?
That's a great question. I think it gets to some of the, gets really at some of the heart of what we're trying to find in the field. So we've looked at schizophrenia, for example, in European populations as well as in East Asian populations. And when we do that, the top genetic variants that are contributing to risk tend to replicate across the different groups. So it's not that like the things that are the most important or significant are not shared. We are finding that those are shared. The problem is when you start to aggregate across many different genetic variants across the genome, which in aggregate contribute more risk than those ones at the very most significant end of the spectrum that are most robustly being identified, then these subtle biases start to creep in and start to get compounded by the fact that we're aggregating across all of these different genetic variants. So also mentioning like the giant study that I talked about earlier for height, for example, if you look at the genetic variants that they identified in that study that were most significant, those were replicating super well across different studies of height. So I see like kind of two big use cases for doing these large genetic studies. One is for risk prediction, which clearly these issues play a huge role in. And then one is for using as a starting point for downstream functional analysis. And for that latter step, these issues of residual stratification and confounding and differences across population are important, but actually don't change which genetic variants we're going to go after. Whereas for the polygenic score side of things, these biases are going to be super problematic when we go to implement these as sort of biomarkers. So it's an interesting question. I really like Dave Curtis's paper on that. It was really interesting.
With that, I'd like everyone to join me in thanking Dr. Martin for her really wonderful talk.